Okay, very well. So what did we do in the last class, Sid? We finished till pleiotropy, right? As I can see.
Yes.
Am I audible clearly and also visible clearly?
Yes, yes.
Yes. Okay. So let's just start from quickly from linkage. Okay.
Okay.
Okay. So tell me, what do you understand by linkage, and just go from here. Yes. Quickly.
Linkage basically means okay. First I tell who described this. T.H. Morgan described what is the linkage for the first time. He found
Yeah.
linkages in Drosophila, that is fruit fly.
Yeah.
He did an experiment.
Yes. Where? What did you do in that experiment?
He took Drosophila like fruit fly, and he hybridized yellow bodies and white-eyed females with brown bodies, the red-eyed male.
Yes, right.
Then and
yes,
they were intercrossed, like F1 progeny, they F1 progeny. Then
yes. So finally, in the F2, yes,
he got the
tell me.
then in F2 generation, he got like the ratio that was not similar to previous one. What Mendel
what do we mean by what do you mean by previous one?
I think previous one was
yes. So this 9:3:3:1 ratio is basically what is followed when you follow the Mendelian rules for inheritance, correct?
Yes.
And he figured out that this ratio was not followed by the progenies of the F2 generation when he crossed a yellow-bodied, white-eyed Drosophila with a brown-bodied, red-eyed Drosophila. Now this tells what he actually figured out was, you can also write down, when I say that it did not follow 9:3:3:1, I mean the parental, write down, the parental combination was more. The parental combination was more inherited, and the recombinants were very less. Okay. Is it clear? There?
Yes.
So these two results actually tell that there was not proper recombination happening, correct, between these two genes. So they are not independently segregating from each other, which is the third law of Mendel. So it violates the third law. That's why it's an exception of the third. Yes, good. Now let's go to this part. Tell me.
Now, chromosomal theory of inheritance was described by Walter Sutton and Theodore Boveri in about 1902. They stated that behavior of chromosomes was parallel to behavior of genes, and used chromosome movement to explain Mendel's law.
Yes. So what is this states in simple words?
So in theory, they describe that the hereditary factors are carried in the nucleus. Nucleus contain chromosomes. Then, like the Mendelian alleles.
Yeah. Okay. It can be even more simple. Just look at the board. It is written. Okay. It states that pairing
the pair and separation of a pair of chromosomes would lead to pairing or separation or segregation of a pair of factors that they carry. that is gene.
Exactly. So simply, if you pair chromosomes, you are pairing genes. If you are separating chromosomes, you are separating different genes, right?
Yes.
Yes, gene copies basically. Yes, continue.
Then the theory united the knowledge of chromosomal segregation with Mendelian principle and inheritance.
Yes.
Mendel published in 1865.
Yeah.
It remained unrecognized till 1900s. Then, because of lack of communication, he was not able to describe it properly what work he did. Then he used, because he used mathematical concepts in biological phenomenon, he compared mathematical concepts with biological phenomenon. At that time, it was not recognized that that mathematical equations can also be used in biology.
That was not accepted. It was not popular. It was not very popular to use mathematics, to use the concepts of mathematics and explain biology. So the contemporary scientists or the biologists of that time did not, most of them did not appreciate Mendel's work. Yes. And third?
Yes. Concepts of factors, that is genes being stable and discrete units present in a pair that never blend, was not acceptable at that time.
Yes.
So three scientists basically gave his work after his death. They were Hugo de Veris, Karl Correns, and Eric von Testermatt.
Sherman. E is
Test.
silent.
Yeah.
You know, you can call him von Schermack or Erin von Schermack.
So basically, they rediscovered. They were rediscoverers of Mendelism. Okay.
Yes.
Now.
And also raised the work to the status of laws, especially Hugo de Vries. Okay.
Now we studied homologous chromosomes. Homologous chromosomes basically are of same length, same short arm. They have one short arm and one long arm length. They have same number of genes present at same locus of the chromosome. Then
yes,
first we did one. We can say we crossed over here. We crossed over.
We didn't crossed over. I explained you that there is a crossing over that happens in homologous chromosomes during the process of meiosis. In this crossing over, like two non-sister chromatids of two homologous chromosomes cross with each other physically and form this chiasmata. Okay, it's called the crossing over bridge. And exchange of genetic material
happens. Yeah.
And as a result, what happens?
We get two different parental combinations.
No, we get two parental combinations, okay, and two recombinants, non-parental combinations.
Yes.
Okay.
Then we did the experiment, that is linkage, based on linkage.
Yeah. So you understood this part. This is from NCERT. We realized that when the physical distance between the two genes is less, there is more linkage and less recombination. When the physical distance between the two genes is more, there is less linkage and more recombination, right?
Yes.
So this cross B actually is following Mendel's law, because here the recombinants are 37.5% approximately, right?
Yes.
Okay, good. Then finally we do two more things.
Polygenic inheritance.
What was this?
Okay. Polygenic inheritance, it is a type of inheritance in which a trait is controlled by three or more genes. Such traits are known as polygenic traits. Okay.
Yes.
Now,
For example, human skin
for
color
example,
was one.
human skin color, yeah. Then
Yeah.
human hair color, okay. Human height,
Not.
okay.
Yeah. Human height is another, and you will see that we also assume that, let's assume that the skin color is controlled by 4 genes, and we saw that how the darkest shade and the fairest
Yes,
shade can come up through genetics. And I also
we did
told
an
you,
example here.
yeah, I also told you that these genes, polygenic inheritance or polygenic traits, are also influenced by the environment. Like in this case, it is influenced by the exposure to sunlight, right?
Yes.
More sunlight, darker the skin, less sunlight, fairer the skin. And the last thing we did was pleiotropy. Tell me.
That is just opposite of polytrophy. Okay. It is a phenomenon in which a single gene exhibits or controls multiple phenotypic expressions. Okay.
Yes.
One gene can, okay. Example, we did diseases, condition, phenylketonuria.
Yes.
A single gene is mutated through an enzyme.
Not through an
Okay,
enzyme. The single gene is mutated, and that gene
mutated
codes for an enzyme. So the gene
coding
is
of
basically for this enzyme. Okay, this is the enzyme ka gene.
okay, phenylalanine hydroxylase, that is
Yes,
enzyme.
that is the enzyme.
Okay.
Yeah.
The affected individuals show like mental retardation as well as reduction in hair and skin pigmentation.
Okay. But what happens during this disease? You didn't tell me that. What is this flow chart?
So actually, okay.
You didn't revise? Shall
Yeah,
I?
I did, but that is like ending of the chapter, yeah.
Yes. No, that is, so I took it before because anyways, phenylalanine, phenylketonuria as a disease is again going to come back in genetic disorders. So I thought
Yes.
if I'm taking it as an example here, it's better that I explain it here. So do you remember this flowchart or shall I explain
Yes.
it once again?
Sorry.
Yeah.
So basically, phenylalanine hydroxylase converts phenylalanine into tyrosine, that is one
Yes.
another amino acid. The
Perfect.
level of sorry, the level of phenylalanine increases in the cell. Then
How?
phenylalanine is
When? Why?
uh,
This happens when the, see, remember this red ink is another faulty pathway.
yes,
If phenylalanine hydroxylase is normal, it converts phenylalanine into tyrosine, right?
yes,
But
but
if
it's
this
not.
gene is
Yes.
mutated, then this enzyme will not be formed. And if there is no phenylalanine hydroxylase, then it will not convert phenylalanine into tyrosine. And as a result, the level of phenylalanine, because it is not being converted now through this enzyme, will start rising in the cell. Once it
Yes.
reaches a very high level in the cell, then without this enzyme, the body tries to deal with it and it cannot deal with it effectively. So phenylalanine gets converted into phenylpyruvic acid if it is accumulated in large quantity. And that phenylpyruvic acid is harmful to the cell, and this causes all these symptoms of this disease. It affects the nervous system, okay, which is the brain, and causes mental retardation. It also affects the skin cell, causes loss of pigmentation. It also affects the hair follicles and causes loss of pigmentation in hair as well. Now, since it affects multiple different characters just by mutation in one single gene, it is called one gene controlling many characters or many systems. Is it clear?
Yes.
Yes. So do you understand that this red thing is when the phenylalanine hydroxylase is not present? Can you see these
Yes.
red marks? Okay.
Oops.
Okay, very well. So today I will start with this another important concept, which is of sex determination. Now we already know about the sex determination in humans, right?
Yes.
So here we are trying to figure out the genetic basis. We are going to study the genetic basis. So you already know the genetic basis for humans. So let's start with the very simple one, which you already know. Human. Tell me, how does in humans sex is
So,
determined? Yeah.
so human sex is determined by XX and XY type.
So in humans, there are sex chromosomes, right?
Yes.
That define the sex. And how many types of sex chromosome can be there in humans?
Two types.
Yes. What are these two
Um,
types?
that is XX and XY.
No,
X
X
chromosome and Y
chromosome,
chromosome. And Y chromosome.
and it can be a Y chromosome,
Hmm.
right?
Yes.
Now, if from both the parents an organism gets X and X in the first case, then it will develop into,
A female child.
yes, it becomes a female. Simple. If in case B, it gets X from the mother and Y from the father, then it becomes,
Male.
which means males are XY and females are XX, correct? This is simple.
Yes.
You can also see here that in case of humans, females are homogametic.
Yes.
What does the word homogametic means?
Both have both are same gametes, X and X. So,
Yes. So produce same type of gametes. Is it clear?
yes.
Whereas in case of males, you can see that males in human males are heterogametic. What do we mean by heterogametic?
They produce different types of gametes.
Produce two types of gametes, not different.
Yeah, two types.
What are the two type of gametes
X
that they
and Y.
produce? Yes. So here, all the gametes will be 22 autosomes plus XX, right?
Yes.
In this case, it can be 22 autosomes plus X. Sorry, I wrote one extra X here. 22 autosomes plus X because it has X and X, right? Oh,
Yes.
just 1%. X. In this case, it could be 22 plus X or
22 plus 1.
22 autosomes plus Y. These are the two gametes that males can form. So males are heterogametic and females are homogametic in case of humans. And this type of sex determination system is called XY system. What it is called?
XY system.
Now this same XY system, write down, XY system is also present. XY system is also present in few other insects, like fruit fly. You know what is fruit fly called?
So Drosophila melanogaster.
Drosophila melanogaster. Okay, just remembering Drosophila is also fine to work with. So you understand XY system, the first system?
Yes.
It's present in humans and it's also present in few insects like Drosophila. Okay. Now, coming to the second type. In more insects, like in other insects, for example, grasshopper and cockroaches, okay, there is a XO system. There also they have sex chromosomes, but there is XO system. So write down. In grasshoppers and cockroaches, in grasshoppers and cockroaches, there is XO type of sex determination. In this, there is only one type of sex chromosome present. Okay, in the species. Is it clear, Zaid?
Yes.
And that one type of chromosome is X chromosome. And the sex is determined by how many X chromosomes. Write down. The sex in these organisms is determined by the number of X chromosomes present. So sex is determined by the number of X chromosomes present. If there is 2 X chromosomes, okay, it will become a
Female.
yes, female. But if there is only one X chromosome, so if there is only one X chromosome, we write O. O means 0 or nil. O is not something. O is not another type of chromosome. O is the absence of any chromosome. Because if we just write X, we have to write something. So nothing came from the other parent. Okay. So this will develop into a male. So if there is only one chromosome, one sex chromosome, which is X, then it forms a male. If there are 2 sex chromosomes, but the chromosome is same. You understand that, Zaid?
Yes.
It is the same chromosome. The copy, the number of it is of deciding whether the organism will be male or female. But you tell me here, still the females are homogamitic, right? Am I right or wrong?
Yes.
Because they can only produce one type of gamete, autosomes plus X. Yes or no?
Yes.
But what will be males? Males here are heterogamitic, just like the XY system. Okay, so if in a species the male produces 2 different kind of gametes, then it is called male heterogamety. And if the female produces 2 different kind of gametes, it is called female heterogamety. Is it clear?
Yes.
So in XO system also, there is male heterogamety. Males are heterogamitic, just like XY system, right?
Yes.
Now, let me come to the third system, which is called ZW system. Okay. Now, this is different. And also the name of the sex chromosomes are different. They are not X or Y chromosomes. And this happens in birds. Okay. For example, your chicken. You know chicken? Of course.
Yes.
So in birds, ZW system is there. And in this, write down.
So does this happen in fishes?
In, sorry?
Fish.
Some, not all. Okay. ZW system. More prominently, we study them in birds, but you are right. But not in all fish. Okay. Fish are very, very like dynamically different in terms of their reproduction. So in ZW system, if it is Z and Z, then it will be a male. You understand?
Yes.
And case A. And if it is Z and W, then it will be a female. You understand?
Yes.
Now, what can you tell about this system? What is different here?
So male is having homolytic homogenetic
Homogamitic gametes. Yes. So here you see that males are homogamitic, not the females. Okay. Males can produce only one type of gametes because they have Z and Z. Both their sex chromosomes are same. Is it clear?
Yes.
Whereas females are producing Z and W. Now, how to remember this? Remember this W here. W is for women. Okay. W for woman.
Okay.
So in this system, how I remember, in ZW system, whoever has a W is a woman, is a female. So if a bird has ZW, that bird is a female bird. And females, hence, in this system are heterogamitic. They produce two different kind of gametes, one containing Z and the other containing W. So in ZW system, who is responsible for the sex of the upcoming offspring? Yes,
Female parent.
female parent. Unlike the other two systems, because there the male parent's gamete is responsible. So write down, in ZW system, the female parent is responsible for the sex determination of the offspring. Okay, not the male parent. Is ZW system clear to you?
Yes.
So in this there is female heterogamity, and in other two, XO and XY, we have male heterogamity. Okay. Now coming to the fourth and the most important, from where we have seen multiple questions in exam, is haplodiploidy system. Haplodiploidy system of sex determination. Now this happens in honeybees. Honeybees, also in ants.
Butterflies.
Okay. And many wasps. Yes, and some butterflies as well. But prominently we'll study in honeybees. Okay.
Yes.
Now before going forward, tell me something about, let me tell you something about the honeybees. So you know that in a honeybees are social animals, right, Seth?
Yeah. Yes.
And honeybees, tell me what do you know about honeybees?
So honeybees basically have, if we talk about their family, then they have a queen, they have drones, and they have drones, and one more is there.
Queen, drones, and who works for the colony? Workers.
Worker bees. Okay.
Yes. So in a honeybee, so honeybees live in beehive, right?
Yes.
We call them beehive. So they are social animals, right? Like ants. Ants are also social animals. Now in a beehive, there are three types of honeybees. First is a queen bee. Okay. What is the gender
Yes.
of the queen bee?
Female.
Queen bee is a female, correct. And queen bee is the only fertile female. Okay, in the hive.
Yes.
So only queen bee can lay eggs. So technically, all the bees in the beehive are kids of the queen bee, right?
Yes.
Then second comes worker bees. Now, worker bees and queen bee is single. One in each hive, in each colony. There cannot be two queen bees in one colony, okay? Only single
Yes.
queen bee will be there. Worker bees, on the other hand, are also females, right? But
Yes.
they are infertile. Okay, they cannot produce offsprings. They cannot lay eggs. The third are the drones. Now, what is the gender of drones?
Male.
They are males. Are they fertile or infertile?
Uh, Infertile. Yeah.
See, if they are infertile, then who will queen bee for reproduce
Yeah,
with?
yeah, they are fertile.
There is nothing like a king bee. Okay,
Yes.
there's only a queen bee. No king is there in bees. So males are fertile. So they produce sperms, and only drones can reproduce with queens. Like drones can only reproduce with queens, not with the worker bees. You understand? So the
Yes.
social setup is clear to you.
Yes.
Now let's talk about the sex determination. Now the sex determination happens like this. Let me start first with a female bee. So let me start with the queen, because queen is the only fertile female, right?
Yes.
Queen bee. Now all the females in a in a bee colony are diploid. You know what is a diploid and what is a haploid situation?
Yes, sir.
Tell
They
me.
have two n number of chromosomes.
They have two copies of their whole genome. Remember, ploidy means
copies.
copies.
Yes.
I always tell you. So queen bee has total chromosome as 32. So and all the females, even the worker bees, have 32 chromosomes. So both queen bee and worker bee have 32 chromosomes, which means they are diploid, 2n. So this is the 2n number. Okay. If this is the 2n number, what will be the n number for this species?
16.
16. What is the 2n number for human species?
2n number. I didn't get your question.
What is the 2n number for our genome? How many chromosomes?
That is 44. That is 44.
46. Our diploid cells have 46 chromosomes. That is our 2n number. What is the n number of humans?
14. Sorry.
23.
26, 26.
Half of 46. So sperms and ovum contain 23 chromosomes each. Then when they fuse together, zygote contains 46 chromosomes, right? Because the nucleus fused together. Is it clear? Said. You have to revise this on a daily basis, okay? Now, if queen bee is a female and she is diploid 2n, then when she will make gametes, the gametogenesis will happen through meiosis or mitosis?
Meiosis?
Yes. Gametogenesis will happen through meiosis. Correct. And if it happens through meiosis, then she will produce from one cell, she will produce two cells, right?
Yes.
and both will be egg cells, right? And what
Yes.
will be egg cell? It will be haploid.
16.
How many chromosomes will an egg cell have?
16 chromosomes.
16 chromosomes. And this 16 means it is n number, right?
Yes.
Same, there will be another egg cell. Okay, let's say. And it also has 16 chromosomes. Now, two things can happen. One thing, first thing is, always remember, whenever there is fertilization, so this egg cell fuses with, write down, if this egg cell fuses with the sperm, now what will be the number of chromosomes or the ploidy of the sperm?
32.
Sperm, how can sperm be 32?
Sorry. Sperm will be 16.
Sperm
16.
cannot be diploid, right?
Yeah.
It is also
Yeah,
haploid,
16.
contains 16 chromosomes. So what has happened here?
They form diploid.
Sexual reproduction has happened. Sexual reproduction takes place. And if sexual reproduction takes place, then all the progenies that are produced through sexual reproductions are females. Female bees. Is it clear?
Yes.
Every time when an egg cell fuses with a sperm cell, undergoes sexual reproduction and fertilization. So this process is called fertilization to produce embryo which is diploid. So females are produced. And how many chromosomes do females have? 32 chromosomes. Yes, always. So this is how females are formed. They are always diploid. So write down, all female bees are diploid. All female bees, and including workers and queen, are diploid. Okay,
Yes.
is it clear? Now
Yes.
let me take another scenario on the opposite side. Let me this time start with a male. So if it is a male or a drone bee, which is a male bee, so what will be the number of chromosomes that it contains? All the males are
16.
haploid. It contains 16 chromosomes. Sorry, I forgot to tell you one thing here. So this other egg cell which is left, if it remains unfertilized, so the one possibility is that it can fuse with the sperm and undergo sexual reproduction. The other possibility is this egg cell remained unfertilized. Okay, if it remains unfertilized, then what happens? It develops into an embryo through parthenogenesis. You know what is parthenogenesis? You study parthenocarpy. Yes, what is parthenogenesis?
So
It is development of embryo without fertilization. Okay, so parthenogenesis happens, and through parthenogenesis, what are produced?
drones.
Males.
Yeah, males.
Okay, so male bees are produced. Male bees. Always they are produced from an unfertilized egg. So they have 16 chromosomes. Okay, which is the haploid number. Is it clear?
Yes.
So if a female undergoes sexual reproduction and fertilization, she will always produce females. If the eggs of that female or that queen bee cannot fertilize and remains unfertilized, then it develops into a male. So to begin with, now you know that males have 16 chromosomes, right?
Yes.
If males have 16 chromosomes, which is already n number, then tell me, if they will produce gametes, the gametogenesis will happen through meiosis or mitosis?
Mitosis.
Why?
They cannot further divide. So mitosis.
They can divide. In order to produce gametes, they also need to equally divide their genetic material.
Yeah.
Remember? And the
Yes.
genetic material is already one copy. Now, it cannot undergo meiosis because that will reduce. Meiosis is a reductional division. And write down somewhere in your copy, only diploid organisms can undergo meiosis. Not haploid, not triploid. Write down. Only diploid organisms can undergo meiosis. Okay.
Okay.
So gametogenesis will happen through, that is the reason that it will happen through mitosis here. That is special. Always we have read in humans that meiosis happens for gamete formation. Meiosis happens for gamete formation. But here it's not the case. So they will form sperms which are haploid, just like the body. So drones are haploid, their sperms are also haploid, of course, right? And
Yes.
sperms have no other option than to fuse with egg cell, another
Yes.
haploid, because sperms cannot form an embryo without fertilization, correct?
Yeah.
Yes or no? So what will happen?
So they will form female worker bees fused with female, I mean queen.
So if this happens, what will be formed? It will be a 2n cell, right?
2n cell.
N plus n will form 2n. And what is a
Yes.
2n? Diploid are always
female.
females. So a male can only form daughters, right? It can only give rise to daughters.
Yes.
It can never give rise to sons. Do you understand this?
Yes.
Yeah. So let me ask you one thing. Can a male honeybee in the beehive, can it have a father? Can a male honeybee have a father?
No.
Why?
So it can only have grandfather.
Yes. Why it cannot have father is the question.
Sorry, forgot the reason.
I just give you the reason here. It cannot have a father because it is produced from a unfertilized egg. If it is an unfertilized egg that is producing the male cannot have a father because there is nothing. There is only one parent involved. So it is like an asexual reproduction, right? Only one parent is involved.
Yes.
Yeah. That's called parthenogenesis. That's why males cannot have fathers. They also cannot have son because the only option a male bee has is to produce sperm, and then those sperms have to be fertilized with the egg cell. And as a result of which, a haploid sperm and a haploid egg cell will give rise to a diploid organism. And diploid in honeybees are always females. So write down, a male honeybee, a male honeybee can never have father and son. Can never have father or a son. But it can have a grandfather. How can it have a grandfather? Can you explain?
So as male produces sperms by mitosis, they fertilize their eggs with uh, they fertilize their n number of
Start from the beginning. Don't complicate. You are
sperms.
just again. How it
Okay.
can have a grandfather? Start.
It can have a grandfather as the male produces sperms by mitosis. So
I'm
so this
asking, when male produces something, it is going to the next generation. You are starting from the male. I'm asking, go back. Don't go after. Male produces sperms, which means you are moving
by
ahead
queen,
from that.
right.
Go back. How can it have a
uh,
grandfather?
The so when can I start from female, that is honey?
Start from the male. Start from
Male.
a male and tell me, how can that male have a grandfather? The male can have a mother, of course.
Yeah.
Males do not have fathers, but males have mothers.
Yes.
And that mother had a father.
Yeah.
Simple. So if a male had a mother and that mother had a father, so male had a grandfather?
Yes.
Yes, simple. Nothing more to explain. Similarly, a male can have a daughter, right? It can only have a daughter in case of honeybees. And that daughter can have further daughters, right? Through sexual reproduction.
Yeah.
So, or sons. So a male can have a grandfather and a grandson.
Yes.
But not a father and a son. Okay, is it clear?
Yes.
Please write this down.
Certain.
Done?
Yes.
Okay, very good. So let me start now. This is sex determination. Any doubt in this? Anything you want to ask? No? Okay, so let me start now with mutation. You know what is mutation?
Yes.
Tell me, what do you know about mutation?
It is like sudden inheritable changes in the genetic material.
Okay, but why these changes? Sudden inheritable changes, you said. I'm asking, are all mutations inheritable?
All mutations are no. All mutations are not inheritable.
Okay.
They can be
They can be
carried out by
carried out by whom?
Okay. All the
How
mutations
are
are not
things
inherited.
mutated? Yes, how are things inherited?
Things are
Through what?
inherited
Through gametes.
Gametes.
Anything that has to be inherited has to inherit through gametes. Any character that the parents want to send their kids, they will send it through the gametes, the sperm or the ovum. So if there is a mutation in the body that does not reach or that does not happen in gametes, will it be inherited?
No.
No, it will not be inherited, correct?
Yes.
Yep. So only those mutations are inheritable which are
which
in the
are
gametes.
in the gametes of the parents.
Yes. So let's first define mutation. Write down. Mutation is the sudden and random alteration or change. Sudden, random alterations or changes in the DNA sequence. In the DNA sequence that results in change in the genotype and the phenotype of an organism. You know genotype and phenotype now, right?
Yes.
Phenotype of an organism. Is it clear?
Yes.
And on the next point, mutations are inheritable to the next generations. Mutations are inheritable to the next generation only if it affects or it is carried by the gametes. Okay,
Yes.
clear?
Yes.
Yes, no doubt, right? Now
Yes.
let's come to types of mutations. So some basic ones I'm telling you for your entrance. Okay, so there can be few types, like multiple types of mutations. The first can be point mutation. What does point mutation means? What is point? One single dot, right? At one single location. So in a point mutation, write down only one single base. Only one single base in the DNA sequence is altered or changed. Okay.
Yes.
For example, let's say this is a normal DNA sequence. Okay, so a normal DNA sequence is this: A, T, G, C, A, T, G, C. This is the normal DNA sequence. Let's assume, okay? And somehow this G gets mutated or changed, and the diseased or the affected sequence becomes A, T. Instead of G, now there is another A, C, A, T, G. So can you see that actually the reading frame, remember I told you there is a reading frame, ATG, CAT, GC, let's say
Yes.
there is a also here. So GCA. This is the reading sequence, right? DNA is
Yes.
read in the codes of 3 bases. Similarly, here also the reading frame is same. 3, 3, 3. So the reading frame does not changes. But what changes is this position, right?
Yes.
This is altered position. There has sorry, not this one. This one. Yes, this is the altered position, correct?
Yes.
Now here it should have been G, but here it is A. This is called it's a point mutation. Clear?
Yes.
Now example of a point mutation, write down. Is the disease called sickle cell anemia. Okay, in humans it
Yes.
happens due to a point mutation. Clear?
Yes.
The second type of mutations, and also write down, in a point mutation the reading frame does not shifts. The reading frame do not shift or changes. Okay, it remains the same. Is it clear?
Yes.
Very good. Now the second kind of mutations are where the reading frame changes. So this is called frame shift mutation. Frame shift mutations. Okay.
Yes.
Now in frame shift mutations, it can be of two types. One, deletions, deletion mutations. Like something got deleted, right? Some
Yeah.
bases got deleted because of which the frame shifted. Or insertion mutations. Okay. Now let's take an example. Let's say there is a normal sequence for case A. Let's say the normal sequence again is ATGC, ATGCA. Okay. Now there is some deletion. Let me write ATGC. Now, because of some deletion, let's say this G gets deleted. So if this gets deleted, what will be the affected sequence or the abnormal sequence?
ATC,
ATC, ATG, CAT, GC, right?
Yes.
Now, if we read the frame now, the normal frame was ATG, CAT, CGA, and TGC, right? What is the frame now? ATC, ATG, CAT, and GC, and something else will be there, right?
Yes.
So do you think the frame has shifted now? This C became the part of the first codon. This G became the part of the second codon, where it should have been the third. This T became the part of the third, where it should have been in the fourth, right? So the whole frame shifted now because of one thing getting deleted. Similarly, if you insert something, then also the frame will get shifted,
So
right?
first I thought like if we delete something like it will break DNA bond will break. Is it
Yeah,
possible?
yes, it always happens during, most of the times these kind of mutations happens during errors in DNA replication. So during DNA replication, the DNA opens, right? You know that the
Yes,
two
yes.
strands of the DNA opens, then once each strand synthesize a new complementary strand, and then hence there are two strands, two separate strands by the end of replication. Okay, makes sense?
Yes, yes.
Yes. So that opening is very, very important. And when you're deleting something, it's like you forgot to put that base while you were making the complementary strand. Okay.
Yes.
Or as you said, there was a cut in the DNA and some bases got removed, chopped off. That can also happen. But either way, if there is a deletion or there is an insertion, in either like either way, there will be a change, shift in the frame, right?
Yes.
Okay. Now write down
Okay.
frame shift mutations are more detrimental. Can you tell me why? This was one question also. Frame shift mutations are more detrimental. Detrimental means damaging or lethal to the cell or to the organism. Can you think why?
Uh,
Can you can you think the logic?
mm,
Simply,
so
when the frame is
maybe,
shifting,
but I don't know the logic behind. Okay. Logic is there, but I don't know the reason.
yeah. So see, when we were not shifting the frame, actually there is just one problem at one position. Rest everything is same, right? So when the protein will be formed, it will be the same protein with some minor problem, right?
Yes.
But here, when the whole frame gets shifted, the entire protein will be different. So you want to make a protein that helps you do an important function in the cell, but instead you are reading the sequence as an entirely different codon, and you're making an entirely different protein, because these three codes give rise to one amino acid, right? You know that.
Yes.
So now there will be a different, it's a complete different sequence of amino acids. So write down, uh, due to frame shift mutations, write down, due to frame shift mutations, a completely different, wrong, completely different, wrong, or non-functional protein chain is synthesized, right?
Yes, sir. It is non-functional means like it is harmful or not harmful.
It does not
Non-functional at
functions.
all.
So
So
you need
it's not.
retinol in your eyes to see.
Yeah.
If the retinol is non-functional, will you be able to see now?
No, sir.
You need melanin in your skin to protect you
Yes.
against UV.
Okay.
If that protein is non-functional, will you be able to? So is
No.
it, so the non-functionality of an important protein becomes harmful to the body, right?
Okay, so yeah. Got
Yeah, you understood?
it. Yes.
Yes. Okay, cool. So this brings us to, okay, one more thing I'll tell you. I don't know, but let me just tell you. Um, Factors that causes mutations So what causes mutations? What is the cause of mutation? So these were the type. What are the cause of mutations? Now this is a very broad topic and we will not discuss it in detail because it's not important for your syllabus right now. Just know that whatever causes, it can be either a chemical factor, right? That can cause mutation, or a physical factor.
So how physical?
Sorry?
So how physical factor?
Yeah, I'm telling you. And in both the cases, it is called, whatever causes mutations are called mutagens. What are they called?
Mutagens.
Yes. Now you ask how physical. So how chemical, you understand? What is the chemical factor? Actually, you should
Yes.
have asked how chemical. So an example of a chemical factor that causes mutation is nitrosourea. Let's say this is a chemical that causes mutation. Physical factor we have always been discussing, UV radiation. UV is not a chemical, right? It is a light. It's a spectrum of non-visible light, ultraviolet. It is a radiation, right?
Yes.
So UV radiation is a physical factor that causes mutation. But whatever it is, whether a chemical or a physical, it is called a mutagen. Is it clear?
Yes.
Okay, very good. So I'll take a quick 2 minutes break here. Okay. And then will rejoin for genetic disorders and pedigree analysis. So that is the last thing that we have to cover in this chapter. Then this chapter will be over, but it is important. So it will take one more class to finish this chapter. Okay. But please revise everything till now. I give you 2-3 minutes. I'll be back in 2-3 minutes. Okay. Said, just a quick break. You can also use it as a water break.
Okay, sir.
Okay, sir. Yeah, I'll be back.
Thank you so much.
Yeah, just revise. I'll be back. Okay, Zed, are you there?
Yes, sir. Yes, yes.
Yes. Done. Revision
Uh,
is done.
so I have a doubt. So,
Yes, please ask me.
uh, why did T.H. Morgan selected Drosophila Drosophila as this specimen for his experiment?
Yeah. So T.H. Morgan, so the reason behind selecting Drosophila was that T.H. Morgan wanted to do the same kind of genetics on something different, a species. So Mendel did it on peas. So there was no point of repeating that whole experiment on peas. And by that time, Drosophila developed as a very good model system to study genetics because Drosophila also has a very short lifespan. Oh, short life cycle. So the life cycle of Drosophila is only 10 days. Okay, the lifespan can be 2-3 months, but the life cycle is only 10 days. So in 10 days, you get another generation. In the next 10 days, another generation. So this is easier to study, right? And Drosophila also had many characters that can be studied, and they are contrasting characters. So that's why he chose Drosophila. It's not like he knew beforehand that if I will choose Drosophila, I will get different results and I will discover linkage and all, right? He just chose that because
Yes.
for the same reasons Mendel chose P. Okay. Cool.
Uh, yes.
Yeah. So it has short lifespan, short life cycle. It gives lot of progenies in one generation, and there are many contrasting characters that can be studied in Drosophila. Easy to culture, easy to self cross, easy to like put a self cross or a cross, opposite cross.
Yes.
Yeah. So all these factors are there.
Okay.
Okay, very good. So let's start with the final part of this chapter, which is genetic disorders, right?
Yes.
Okay, so genetic disorders. Now, before I tell you about genetic disorders and how are we going to study it, let me tell you about first thing, that is pedigree analysis. So we study genetic disorders, and then for the sake of studying it genetically and what is causing that disorder and the inheritance of it, especially, we use pedigree chart or pedigree analysis. You know what is pedigree analysis?
So it's like basically a doctor that is he he or she gives the patient or the mother female uh, pre pre you can say a chart. He makes a chart on the base whether or not his child or her child will get any mutation in her or his gene. So
Yeah, through pedigree analysis we can predict. That's true. But
yes,
what is pedigree? It's not like only a doctor can make a pedigree. You can also make a pedigree if you understand the medical condition of all the members of a family. So a pedigree analysis is just like a family tree. Have you made a family tree? Is
yes,
a, we make our grandparents on the maternal side, on the paternal side, then our parents, then they get married, and we form us and other siblings, right? That is a family tree,
okay,
generations, right? Basically. So
yes,
pedigree analysis is just the same. In pedigree analysis, we are studying certain traits or disease through symbolic representation of individuals across generation. Is it clear?
yes,
So write down pedigree analysis is the study of pedigree analysis is the study of family history family history. Study of family history about the inheritance of a particular trait or gene. Particular trait gene which is responsible for any disease or disorder, genetic disease or disorder. Is it clear?
Yes.
In pedigree analysis, write down in pedigree analysis, symbolic representation of normal carrier or diseased or affected individuals is done. Okay, is it clear? So we symbolically represent, we use symbols to represent different type of individuals through generation. Is it clear? So it is basically a study of family history through generations with
Yes.
respect to any particular trait or character that we want to study. Okay, Zaid? You understand? Am I audible?
Hello.
Yes, Zaid. Am I audible now?
Yes, yes.
Yeah, you lost connection for some reason, I think.
So
Okay,
I
so I
got
was
the
asking.
points. Okay. Yeah.
Yes, you understood that what is symbolic representation
Yes,
through
yes.
generations. Now let's figure out what are these symbols. So the symbols used in pedigree analysis, write down symbols used in pedigree analysis. So symbols that are used are, first, if we are talking about a male, then we make a square. Okay, square is for a male, to represent a male gender. Okay, and
Yes.
if we are representing a female, we make a circle. Okay,
Yes.
and if the sex is not specified, so you know that there is an individual here. So for example, you know that your great grandfather had a sibling, but you do not know that whether that sibling was a male like a brother or a female which is a sister. For example, we are making a pedigree chart for someone and we do not know. We know that there were 3 siblings we know that two were brothers. The third was a brother or a sister. It's not clear. So in that case, if the sex is unspecified, we make a diamond. Okay, not a square, not a circle, a diamond. Okay.
Yes.
So this means sex unspecified. Okay, cool.
Yes.
Now, if we have to show, so these are normal. If square, circle, or diamond is there, it is normal. If any of this is colored, like if the box is a solid box, it is colored. So normally it should be colored with the red color, but in exams you will get it black colored. So if this is there, or if it's a circle that is colored, or if that is a diamond that is colored, then what does this means? These symbols means affected individual, affected or diseased. Is it clear?
Yes.
So this is the main thing. Now, another thing is, if we connect a male with a female with a single line, what does this mean? This means mating. They are mating pair, or they are married to each other. Okay.
Okay.
And if instead of one line, there are two lines between a male and a female, what does this means? This means mating. Yes, tell me. Or marriage.
Between relatives.
Yes, between relatives, which means the ancestors share blood. So like close cousins, first cousin, second cousins. So if there is a marriage between cousins or between relatives, then that is called that is represented by a double line. And this kind of marriage is known as, it has a name, it's called consanguinous marriage. Consanguinous marriage. Okay, is represented by a double line. Is it clear, Zed?
Yes.
Now, through pedigree analysis, we can also get to know of the series of the birth. So let's make a pedigree analysis. So if this is a pedigree, so there is a male and a female who got married, and they gave, so they have one son, and they have one daughter, and they have another son. So what does this represent? This represents that these are parents. Okay, and these are offsprings or kids, the first generation, F1. Okay, so parents are always above and kids are always below, linked through this line like this. And the series of birth is from left to right. Write down, from left to right is the order of birth. Order of birth, left to right. So if I mark the kids, this is 1, this is 2, this is 3. Who is the eldest? 1, 2, or 3?
1.
Yes, and 3 is the youngest. Okay.
Okay.
You understood this thing?
Yes.
Now, so you tell me what is the meaning of this pedigree now? If I say, if I make a pedigree like this, there is a male. You have to tell me everything about this pedigree. Whatever you can figure out, you have to tell me. Yes, tell me. This is the pedigree.
Okay. Parents with a male child affected with disease and two female childs and unaffected. Then we can say from here that
No, you
the
don't have to
female
read the
is a
pedigree. You don't have to read the pedigree. You just have to tell. So you forgot to tell me. So these are two parents who
parents.
seems normal, who are not affected. They may be carriers, we do not know yet. Depending on what
Yes.
kind of disease, what kind of genetic disorder it is, they can be carriers. We are not sure of this yet. But there are two parents who seem normal, like who did not have any symptoms for the disease, but they had three kids. The eldest is a daughter, and the youngest is also a daughter. And the middle child, the second born, is a son. The daughters are both normal, and the son is affected according to this pedigree. You understood how to read a pedigree? What I was asking?
Yes.
I'm not asking. So you can write here. This pedigree tells parents with three kids, two unaffected daughters, and one affected son. Okay.
Yes.
Is it clear?
Yes.
Okay. Great. So also one more thing, let me tell you. If any of this symbol, whether it is this symbol or it is this symbol or it is this symbol, unspecified sex, male and female, right? If they have any number written in it, let's say 3 is written like this. So this means that unaffected offsprings, but 3. So this means 3 unaffected offsprings whose sex is not known. This means 3 unaffected offsprings whose sex is not who are male, and this means 3 unaffected offsprings who are all female. Is it clear?
Yes.
So 3 unaffected offsprings. It means okay, understood? All the symbols you have drawn.
Yes. So, but how this will be used in the chart?
It's not used basically, but in like it will not be used in your chart, but sometimes just to save time, if you know that a couple had three kids, all 3 are daughters and all 3 are normal, so you can just, for the sake of representation, make a circle and write 3 in it. Okay, but it's not used in pedigree, but just know it. Okay.
Okay.
Yeah. Okay, cool. So pedigree is clear to you?
Yes.
Now let's come to genetic disorders and write down that genetic disorders can be of 2 types. First is Mendelian disorders. The second is non-Mendelian disorders.
So
Now,
can we say like chromosomal disorders?
yes. So Mendelian disorders, yes, they are at the level of genes. So they are basically gene mutations or gene alterations. So whatever happens in gene, because the inheritance of gene follows the rules of Mendel. Mendelian inheritance happens for the gene, right? Because Mendel only talked about factors which are genes only, right? So his work is on genes. So genes inherit through Mendelian laws, so it's called Mendelian disorder. But chromosomes, if the disorder is at the level of chromosomes, so they are also known as chromosomal disorders. Is it clear to you?
Yes.
So first, we'll start with Mendelian disorders. Mendelian disorders. Now, Mendelian disorders again can be of two types. A, they can be autosomal. So the gene that is affected or mutated, it is on an autosome, chromosome number 1 to 22. Okay, you understand?
Yes. Yes.
Or it could be sex linked, which means the gene that is mutated is present on the X chromosome most of the time. So sex linked, you can also say there are two sex chromosomes, X and Y, but on Y chromosomes, the genes that are present are mostly to give rise to the masculine body, to decide the male gender. Okay, so sex linked disorders mostly by large are on X chromosome, so they are also called X linked or sex linked. It's the same thing. Okay, and for both, whether they are autosomal or sex linked, they can be either dominant or recessive. Is it clear? Because
Yes.
alleles are either dominant or recessive for both. Do you understand this?
Yes.
So there are four possibilities now. A Mendelian disorder can be an autosomal dominant, autosomal recessive, or it can be sex-linked dominant and sex-linked recessive. Is it clear?
Yes.
So write down. There are four possibilities for any Mendelian disorders: autosomal dominant or autosomal recessive, sex-linked dominant or sex-linked recessive. So let me make it like this so it becomes more clear. So it can be autosomal dominant, some recessive, or it can be sex-linked dominant or sex-linked recessive. Is it clear, Seth?
Yes.
So let's start with a few examples. The first one that we will do is color blindness. Let's start with color blindness. Okay, what do you know about color blindness? For every disorder, we have to know four things: whether it is autosomal or sex-linked, or recessive or dominant, what is affected, what gene is affected, and if how, like the mechanism part is available, then that. Okay, and then we'll do some pedigree analysis or pedigree related questions for all these disorders. Now write down, color blindness is sex-linked, which means it is on which chromosome?
X chromosome.
Yes, and recessive. So it is a sex-linked and recessive disorder. Now, the interesting thing about sex-linked disorders is how many X chromosomes do females have? Human females?
2.
And how many X chromosomes do males have?
What?
One. For a sex-linked disorder, the gene has to be present on X chromosome. And for
Father,
it,
father.
sorry?
I think only father. If it is present only father, then only the disease can be transferred to another progeny.
No, that's not true. It has to be on X chromosome. Even if it is on mother, it will be transferred. But the point here is that mothers have 2 X and father has only 1 X. And for any disease which is recessive, any recessive trait, when does it express itself? When we started the chapter, we started it with law of dominance and recessive, dominant and recessive traits, where we discussed that recessive traits can only express themselves in what condition? Go back. I'll not give you the answer. Go back to your notes and figure out when does recessive traits express themselves? Are you going back?
Yes.
Basically, I'm asking whether they express themselves in a heterozygous state or a homozygous state. Recessive disorders.
So heterozygous.
So you mean to say if it is a capital T and small t, it will be dwarf.
No.
Is it possible?
No, sir.
It's not possible.
Homozygous.
So a recessive trait can never express itself in a heterozygous condition. When can a recessive trait express itself?
In homozygous condition.
In a homozygous condition. So for a female to be colorblind, that female needs to have a faulty gene on both the X chromosomes, right?
Yes.
But for a male to be colorblind, because the male does not have any other second X chromosome, even if that faulty gene is present on one X chromosome, the male is affected. The male is colorblind. So recessive disorders, if they are sex linked, then they can also express in a male, even in like in a condition where there is only one X affected. Okay, you understand?
Yes.
Yeah. So this means that males can never be a carrier for a sex-linked disorder. They are either affected or normal. Make sense?
Yes.
So write down. For any sex-linked disorder, especially sex-linked recessive, because if it is sex-linked dominant, then even the females are not carrier, they are affected, because dominant diseases can express even with one copy faulty, yes, in a heterozygous condition.
Yes.
Understand? Yeah. So write down, for a sex-linked recessive disorder, a male can never be a carrier. A male can never be a carrier. Either the male is affected or normal. Okay, only a female can be a carrier for a sex-linked recessive disorder. Only a female can be a carrier for the sex-linked recessive disorder. Now, what happens in colorblindness? Colorblindness means simply the human is not able to see colors. Now, how do we see colors, Zed? What helps us in seeing colors?
We see colors that we have cylinders in our eyes.
Sorry, what cells?
Cylinders. known
there
as
are
color cylinders.
are you saying rod and cone cells?
Yes, yes.
Yes. So in the eye, we have two type of cells, photoreceptor cells called rods and cones. Okay.
Yes.
So inside the eye, there are two type of cells. One is rod cells and the other is cone cells. Now rod cells, remember, they help in low light vision. Okay.
Yes.
And cones help in daylight vision. And when can we see colors? In the dark or in the light, in the presence of light? When are we able to see colors properly? In low light or in daylight?
Sir, daylight.
Yes. So cone cells also give color vision. They are responsible for us perceiving colors. So we can perceive colors because of the cone cells. Okay.
Okay.
Rod cells are only telling about the presence of something. So has it happened to you that in the night, because you know your room properly, so in the night sometimes even if you wake up in the dark and the room is, there is very less slight light coming from somewhere, little light, and in that low light vision you can actually see that there is a almirah there, but you cannot see the color of it. You can just see that there is a darker shade there.
Yes.
There is a shape of the darker shade which is in the shape of almirah. So you know that the almirah is there, but it looks darker and the other things in the room looks lighter. So it's just the low light vision. There is no color associated with that. You understand?
Yes.
Whereas the cone cells are of three types. One cone cells which perceives red, one cone cells which perceives green, and the third cone cells which perceives blue. RGB, red, green, and blue. What are RGB? They are called primary colors. You know that? You can make any color by mixing the primary colors. If you have done art and craft as a child, you know that red, green, and blue is enough. If you have them, you mix them
Yes.
in right proportions and you can make any color, right? Not white, apart from white. In case of color blindness, write down the red and the green cone cells. The red and the green cone cells. Red and green cone cells are affected. Okay.
Yes.
Are affected. So there are genes that help us to distinguish between red and green. Okay. So write down the person. Write down another point first. This defect is due to mutation in some genes. Mutation in some genes associated with red and green cones present on and are present on and are present on X chromosomes. X chromosome. Is it clear?
Yes.
So what makes us see colors, that gene is present on X chromosome. Those genes are present on X chromosome. Now it's clear that if a male, because males have only one X chromosome, so all our color vision is coming from the gene which is present on that one X chromosome. And if that gets faulty, then we will not be able to see color because there is no other X chromosome to rescue it, right? To
Yes.
help us. Okay, good. Now, because it is a X-linked disorder, what is seen is that it is rare that a color blindness happens in females. Write down this question. It is a board's question. Why colorblind females are rarer than colorblind males? Why colorblind females are rarer than colorblind males? Can you think of one reason?
So they become carriers as
I'm not asking that. I'm asking why colorblind females, we are talking about affected now. Why affected females who are colorblind are rarer? Why there are very few females who get affected for colorblindness and many males who are affected for colorblindness? What do you think can be the reason? What it takes for a female to be colorblind? Tell me.
so maybe genetic reason.
Of course, it will be a genetic reason, but what genetic reason? How many X chromosomes do a female have?
So I don't know.
How many X chromosomes do a female have?
2 X chromosomes. Yeah.
Yes, 2, X and X. And for a female to be colorblind, she has to be X colorblind and X colorblind, right?
Yes.
For a male to be colorblind, it just has to be one X colorblind and one Y, right?
Yes.
To get both the X's as colorblind, the female needs to have a father who was colorblind and a mother who was at least a carrier, right?
Yes.
Which is rare to happen. So write down, for a female to be colorblind, she needs to have a colorblind father. She needs to have, or I should draw it like this. She needs to have a colorblind father giving one X carrying the mutation and at least a carrier female, carrier mother, at least a carrier mother to give another faulty X. This is the only possibility, right?
Yes.
So the chances of a colorblind father, a colorblind person marrying a carrier woman or a colorblind woman are very less. So that's why, write down, only 0.4% of the females in the world, 0.4, around 0.4% females have color blindness. And it is not a lethal disease. If you are colorblind, you still live, right? So
Yes.
it's not lethal. Write down. On the other hand, for a male to be colorblind, he just needs to have either a carrier or an affected mother. Either a carrier or an affected mother, right?
Yes.
So if it is XC and X normal, or it is X colorblind, X colorblind, in both the cases, whether it is a carrier mother or it is an affected mother, in both the cases, the male has chances of getting a faulty X. And this is not very rare, right? It can happen, right?
Yes.
It can happen more often than this thing. So that's why male are at disadvantage when it comes to sex-linked recessive disorders, because they only with one faulty copy can become diseased. So write down, around 8% of the males in the world have color blindness. Are you able to distinguish between green and red, Zaid?
Yes. Yes.
This is green, this is red.
Yes.
Right? Yes. So you
Yes.
do not have a color blindness. So 8% males and only 0.4% females. It is around 20 times more, right?
Yes.
Males are affected 20 times more than females for color blindness. Is it clear?
Yes, sir.
So will you be able to answer now for color blindness everything that can be asked?
Yes.
Okay, so your homework is that before you come to the next class, you have to read other diseases. So other Mendelian diseases include hemophilia, your NCRT, sickle cell anemia, and phenylketonuria, which I have already discussed, and thalassemia. These 4 things you have to study on your own and come back. Write down thalassemia, hemophilia, phenylketonuria, and sickle cell anemia. Okay. Then in the next class, we'll finish this chapter and we'll start with chapter 6.
Okay.
The next class, yeah. So come prepared. Zayed, do you have any doubts? Otherwise, I will just finish the class. Any doubts?
So what is the test cross? Just can we do a little summary?
Yes. In brief, a test cross. Let me write it as your doubts. As the name suggests, it is to test. So a test cross is to test whether the whether the dominant parent or the dominant phenotype, write down, a test cross is to figure out whether the dominant phenotype is heterozygous or homozygous. A dominant phenotype is heterozygous or homozygous by crossing it with a homozygous recessive individual. Now let me give you 2 scenarios. Let's say there is a tall plant, okay, Zaid?
Okay.
Now two scenarios where it can be tall is this. A can also be tall and B can also be tall, right?
Yes.
But A is heterozygous tall, right?
Yeah.
and B is homozygous tall.
Mm.
We call it pure line, we also call it hybrid. But just by looking at the plant, you will not be able to figure out whether the plant is heterozygous tall or homozygous tall, because tall is same. Both will look tall, right?
Yeah.
So how to figure that out? So for both, you cross it with a homozygous recessive individual. Okay.
Okay.
So the moment you do this, because homozygous recessive will be short, and short is visible, right? We can just see the short. Whenever we see a short plant, we are sure that it is recessive and it is only expressed in a homozygous condition. So we know just by looking at the plant its genotype if it is recessive, right?
Yes.
In this case, in the F1 progeny, what will happen? There will be four
Oh,
things. Let's say
yes.
capital T, small t, yeah, and here small t, small t. Okay, what will be the offsprings? Tell me.
Capital
Capital T, small t, capital T, small
capital T,
t,
small,
small t, small
small,
t,
small,
small t.
small T.
So do you see
The
that
ratio is
50% 50% are tall and 50% are short, right?
Yes.
But let's see here. What happens here? Here, when we do this cross, it will be capital T, small t, small t, right? What will be the progenies? Capital T, small t,
Yeah.
capital T, small t, capital T, small t, capital T, small t. What?
So
No 50%. All are tall.
1:2.
There is no 1:2 anything. All are same.
All right.
So all are tall. Here,
Yes.
50% are tall and rest 50% are short.
Right.
So just if you do a cross of a tall plant with a short plant, okay, tall
Okay.
plant with a short plant, and if in the next generation in F1 you get half tall and half short, then it means that the tall parent was heterozygous.
Okay.
So this tells tall parent was heterozygous. And if you cross a tall plant with a short plant, and in the next generation all are tall, then it tells that the tall parent was homozygous, right?
Yes.
So this is called a test cross. It is to test whether the dominant trait was heterozygous or homozygous. You understood?
Yes.
Is it clear, Zaid?
Yes.
No doubt.
Yes.
Okay, so I'll see you in the next class then. Okay, take care. Please read and finish this chapter from your side from the NCERT. In the next class, we will start with chapter 6. Okay. Did you send me something regarding the syllabus completion thing, right?
Yes.
What was it for? Is it like
So
you have
that's
been given by school or something?
yeah, given by school.
Okay, I understood. Okay, so we are at the same